Hi, my name is Elissa Wallach, and a couple months ago, my grandfather was hospitalized due to becoming very weak and ill. After many tests, the doctors finally came up with a diagnosis that included MRSA. MRSA is an acronym for methicillin-resistant Staphylococcus aureus. This infection is rather common in hospitals, nursing homes, and other healthcare facilities. After looking into topics for this research project, I found that Staphylococcus was related to our topic of metagenomics. To understand metagenomics, you first need to understand microbes. Microbes is a term used to describe microorganisms such as bacteria, fungi, viruses, algae, protozoa, and archaea. Microbes are so small that they cannot be seen by the naked eye, but they shouldn't be underestimated. They live in communities and work together to make a big impact. They are needed for every process in the biosphere and are found in the human body. Microbes serve humans by producing drugs, fermenting food, and maintaining our health. However, there are some microbes that can be harmful to our health. Staphylococcus is a group of bacteria that can be found in humans, livestock, and other animals. Because it is a bacteria, it is also considered a microbe. There are more than 30 types of this bacteria that result in infections. Staphylococcus aureus is the main cause of most infections that take place in the human body. Diagnosis includes obtaining a culture of the infected area. Most microorganisms cannot grow in a pure culture, which results in a full spectrum of microbial diversity not to be captured. Metagenomics is a genomic technology that allows for a culture-independent insight of microbial communities. Instead of focusing on the individual, it focuses on the community, which gives a much more refined view. Metagenomics has been able to solve the taxonomy up to species and show that various body types acquire different Staphylococcus species. I found that metagenomics can be broken into 4 different steps, the first being genetic material extraction, the second, genetic material manipulation, then followed by library construction, and the final step is DNA analysis. During genetic material extraction, DNA from the environmental sample is isolated. In genetic material manipulation, restriction endonucleases cut the DNA into particular sequences of base pairs. The extracted DNA from one cell is introduced into model organisms that are able to grow, be studied, and express the extracted genetic information in a lab. The DNA material is then replicated to form a colony. Since it's an exact copy of the one strand, it is said to be cloned. The samples are called metagenomic libraries since they consist of metagenomic DNA samples. The final step is to assess the DNA and attempt to answer the question of the research project. So while looking into Staphylococcus, I came across this study called the Single Species Metagenome Subtyping Staphylococcus aureus Core Genome Sequences from Shotgun Metagenomic Data. This paper was published by 6 scientists in 2016. It's a 28-page scientific paper with a lot of overwhelming information, so I broke it apart piece by piece to try and make sense of it. I found that instead of extracting the DNA, manipulating the genetic material, and constructing a library, these scientists skipped those steps and found the needed DNA information off public databases. Metagenome sequence data means that they didn't collect the data on their own. They obtained the data they used from 2 websites that had the desired sequences of Staphylococcus aureus. This is similar to some of the websites that we used in genetics lab. One of the sites that they used was the Human Microbiome Project. The Human Microbiome Project was established in 2008 with the mission of generating resources that would enable the comprehensive characterization of the human microbiome and the role in human health. By playing around on the website a little bit, I found that, let's see here. All right, there we go. I found that there was over 32,000 samples, and there was also From those samples, there was also over 118,000 files that you can search through for these sequences. I also found that you can filter searches of your sequences based on body site, and this is actually what my scientists did in my paper that I read, and I'll explain that later. The second database that they used was called the Metasub Analysis of the New York City Subway. Let's see. This one, sorry. In 2013, the Mason Lab began a project called the Pathomap with a goal to profile the New York City metagenome in, around, and below New York City on mass transit areas of the built environment, focusing specifically on the subways. So this is the website that they went on to do that, and from here you can do a lot. You can look at their projects, and actually they started this project in New York City, and they've actually gone on to do lots of other cities to find their microbiome, as you can see across the world. Okay, so from these databases, the scientists in this study I read about were able to filter out Staphylococcus aureus to learn about genetic variation and biogeography through a long and drawn-out process. Since the process was rather complex, I will go ahead and skip over to the results. As mentioned earlier, the Human Microbiome Project was filtered based on body sites. This figure shows the positive body sites where the Staphylococcus aureus was found. The top 3 places on the body that were sampled had positive reads were the hard palate in your mouth, the left retroarticular crease, that's like on your left side, your ear, right behind that crease on your ear, and the right antecubital fossa, which is the right elbow crease. All 3 of these body sites had positive samples 100% of the time. From this website, different mixtures of subtypes and different human body sites were observed. This figure shown in the paper showed the distribution of Staphylococcus aureus subtypes identified in the New York City subway metagenome data. Box A in the top left corner shows all sites sampled in the central Manhattan area. Box B in the top right box shows sites containing any Staphylococcus aureus reads. And then box C and D in the bottom row have different subtypes of Staphylococcus aureus. From this study, we gained an overview of biogeography of Staphylococcus aureus subtypes from environmental metagenomic data. From these results, other factors such as health insurance coverage, gender, age, tobacco use, surgical history were observed to test if they had correlations to staph infections as well. It was concluded that age, breastfed or not, tobacco use, history of surgeries, and gender were not significant predictors for the detection of any Staphylococcus aureus subtypes. One thing I did find interesting about the results that they were able to conclude was that there was a lower detection rate of staph infections among subjects without health insurance. In fact, the health insurance p-value corresponds to a 50% lower risk of carrying the bacteria among the uninsured. This is hypothesized to be due to the uninsured having less contact with the medical system. The scientists found that 2 of the main challenges in their experiment were missing loci and mixed strain compositions. Missing loci occupied when coverage was too low to guarantee the presence of a read containing a given sequence in the target genome. The mixed strain issue means that the scheme cannot assume that only one subtype is present. They used a software known as binstrain that helped resolve these challenges. I'm going to make an assumption that because we are all in this genetics class, you will someday have a career that will involve science, whether it be in a lab, a doctor's office, or in the outdoors. Metagenomics is a growing field, and there is a very good chance that at some point our work will include metagenomic testing. Metagenomics has the potential to advance many biological understandings for both ecological biology and cellular biology. All life on Earth relies on microbes, so it is to our benefit that we understand them to the fullest potential. By studying microbiomes, we have the potential to cure diseases, clean oceans, and make advancements in agriculture, along with several other positive impacts. It truly is an exciting time to be involved with science.